Quello in alto, dai. Mettiti vicino al microfono, ti va bene? Dimmi come posizione.
So, good afternoon everyone and welcome to this plenary session. I'm very happy to introduce Professor Micaela Liberti, who's a researcher in the field of bioelectromagnetics, and she's professor at La Sapienza University in Rome. And her research interests are related to biomedical application of electromagnetic fields, including electric and magnetic simulation of the nervous system, microdosimetry, and exposure system and patient dosimetry. She's involved in the bioelectromagnetic schools Alessandro Chiabrera since 2004. She has been the director. And she's a member of the Scientific Council of the European Bioelectromagnetic Association. And let's say since 2021, she has been a member of the Scientific Expert Group of URSI and IRP and the Technical Advisory Committee of the International Union of Radioscience, so URSI Commission K. And in 2022, she joined the TC28 of the IEEE Microwave Theory and Technique Society, MTTS. And finally, she was elected as the president-elect of the BioEM Society in June last year. So thank you, Micaela, for being here, and the floor is yours.
So thank you very much for this introduction. I want to thank the organizers for inviting me in that will be sort of the frontiers between the different commissions. And this is me, but what we do would not be possible without a group. And so I would like to introduce at first the group itself. We work at Sapienza University, the Faculty of Engineers. I think some of you have been there in recent years. All the community, it's very active, and some of you should have been here, not here, but in Sapienza for the last General Assembly in 2021. This is the group, and we are distributed in more projects and kind of approaches to this field that from one side it's very narrow with respect to the whole electromagnetic spectrum of research, but still there are many kinds of different applications. They span from basic and technological research, so studies to really understand the mechanism of interaction between biological systems and electromagnetic fields, but then all the possible application of it that we can consider Earth protection. We can hear, all of us can hear a lot about the possible adverse effect of these electromagnetic technologies that we study and apply and implement on the human health, but there, as usual, there are also possible applications. There are biomedical applications that we can distinguish in diagnosis and therapy. So all this is, all the group that I showed before can be in place only if all this is translated in the projects that are the logo that you can see on the sides. So we already mentioned the two possible aspects of this bioelectromagnetic implementation that are mainly coming from different kinds of sources that are typically sources unintended to which population is exposed, like all the telecommunication sources, but also low frequency that sometimes is not the focus of our attention, but don't forget about power lines, and I think you all have heard about the possible effects of living close to power lines. And this kind of research finds the best application in which are the regulatory issues and as a consequence the policies related to that. On the other side, we have what are really intentional sources. There are those that you can find on the other side. They can be sources like coils for magnetic fields, but also different kind of distributed sources that try to use that kind of phased array technique just to focalize in specific parts of the brain. And then we all know about the techniques of magnetic resonance applied for diagnosis. And of course, the applications are, as usual, new diagnostic techniques, but what is even a great focus nowadays is new treatment for that. In the middle, what we see is the human body, of course, but also what I would like to point since the beginning is the fact that when we consider the interaction between electromagnetic fields and the human body, we should always consider that we need to deal with the multi-scale problem that is from a space point of view. So we start from what we call the nanoscale, and we arrive, let's think to our cells that are microscale. Then we have organization of the cells in part of tissues with their details. Then we have organs, and in the end we have the entire body. And we cannot disregard the knowledge of this. But each of these space level has its own time scale that is, of course, very fast for molecules, and as we go up, we can see the timing would increase. And we know this because we know that our physiological functions are much, much lower than the response of molecules. This is what we can intend because now this is the era of nanotechnology, but if we really think about this, let me say that maybe nature is the first real nanoscale architect, because the human body is founded on the organization, as we said, of organs, tissues, but inside the tissue we have specific cells for all tissues, and depending on the kind of tissue, the organization of cells can change. As we can see here, the image is not beautiful for skin, but we all know the fact that skin is an organ. It's very extended, but it's an organ, and it has a specific organization. And we are speaking about new frequencies in telecommunication with higher frequencies, and this means that the absorption will be more and more superficial. And so nowadays, skin is very well studied to understand which can be the effect of electromagnetics on the skin. But the kind of organization of cells inside the skin, it's very peculiar, it's different, they are very compact. they are different, but still each of them are cells. So we cannot disregard how the cell is organized. Cell is not just an envelope with something inside, because what is inside the cell permits and facilitates the function that goes from regeneration to any kind of functions that is the basis of our living. And the basis of the cell is again a nanoscale organization. This is nanotechnology. Let's go to see what this is an image of the membrane of a cell, but inside the membrane we know that the membrane is organized in all these specific molecules that are phospholipids, and phospholipids from a physical description are super interesting because they have this sort of red part is hydrophilic, and these yellow tails, they are hydrophobic. And this is the reason of these apertures. So this is the multiscale from a space point of view. So what we follow in bioelectromagnetic community is what we call multiscale. We already discussed it, multiphysics and multidisciplinary, because I'm sure that many of the aspects that I mentioned you have heard, but some other no. Because when you deal with this kind of interaction, you have to consider all the specific know-how that goes from biologists, then there are medicals, then there are policymakers, and so on. So, what is even very important is to consider that each level of this multiscale has its own proper physical description and possibility to, and the need, not only the possibility, to have an experimental validation of which is the physical description. So we can observe two paths. One is more theoretical and numerical, and the other is experimental. And we need to go through the biological scale. If you think to drugs or any kind of medical application, you know that all the story starts from proving the effect on the cells, and once that is proven on the cells, then people who put this in the market, drugs or biomedical applications, need to prove it in animal laboratory studies, and then there are many levels of statistical power on human cells. They start with few volunteers proving tolerability and safety, then they scale up, and only when the statistics is very large, they can put a drug or a biomedical application in the market. So at any scale, we need both description, understanding from one side, and experimental validation from the other side. But what is even very interesting is that these two aspects, proven at each level, in nowadays are more and more tied together. What is one of the great breakthrough that we observed with genomics around 20 years ago has been that all the scientists have realized the importance of keeping these two aspects, theoretical and experimental, very close together. What you can find here is a readaptation of the well-known Kitano cycle, and he has been the one who first had pushed forward these two terms, dry experiment and wet experiments. And both of these two kind of approaches have close connection to the two parts. So, for example, we can say that we start from some unresolved issues, and we need to model which can be the solution. Then we need to go to dry experiments, but dry experiments rely on models models that we need to simulate. We need to do the simulation to understand which is the result. And even in this case, also dry experiments, we all know that can give us many data that we need to analyze. And on the basis of this analysis, then we can formulate some more hypotheses, we can go to prediction, but then there is the other great aspect for us, who are all physics and engineers, that is the next step. And this is always a technological step. We go towards design of experiments. And designing experiments very often means put in place new technology. But after that, the new technology has been put in place, what we have to do is the wet experiment. And that are more tricky, because in particular in biology, this means that we need to dirt our hands, because there are very often experiments that must be redone, culture that have been contaminated, some experimental mistakes, and so things take a lot of time. And for this reason, it's very important to have always the possibility to check also your dry experiment results. And then there is the experimental data analysis that bring to confirm or reject some hypotheses, but what we know from science is always that some new possibilities are put on the table. And so the cycle starts again and again. And this is not, as been said by us, but again, this is something that has been really pushed by people who worked on genomics around 20 years ago. So this 3M methodology, we start from a description of dry experiments. Why do I want to start with a dry experiment? Because studying the very first level, the molecular one with dry experiments, is what can suggest us where the true interaction between the electromagnetic fields and the biological system can take place. And so we study there the mechanism in order to be able to correlate the mechanism with the evaluation of, again, many of us are engineers, we know that the most important aspect is always to quantify. We rely on numbers, we rely on values. So we need to correlate the effect, the mechanism, to the value that can start the effect, because we need to know when. Both in the cases we need to protect human health, or that we want to interact in order to drive, for example, to drive a therapeutic application. So this is what happened. We study with molecular simulation the mechanism, then we need to correlate this response with a specific value, and this happens at the cell level, and this is what we call microdosimetry. But then we need to scale up more cells or organizations of some tissue details, that is what is called mesodosimetry. And finally, when we are at the main level, at the whole organism, then we speak about dosimetry. So we need to go from nano to macro. This is clear for what I said until now. So when we start at the nanoscale, we speak about femto to nanoseconds, and we go from nanometer to hundreds of nanometers. And all this we describe with discrete physics. But then when we go up, the physics that we all are used, in spite of the topics that I have seen in this program, the physics that we are more used is the continuum physics, when we deal with classical electromagnetics. So this is another important point. We need to bridge this gap between discrete and continuum physics. When we speak about discrete physics at the level, the kind of nanoscale can be approached with very few atoms, with an ab initio description with orbitals and electrons and so on, or they can be done with what is called full atomistic. That means that we only use classical physics, and this is written by hand by the Nobel Prize in 2013, where all the possible interaction potential among atoms describing single molecules are taken into account. But at the very end, the kind of force that is described is a sort of Newton law, where all the atoms are put together. So we speak about hundreds of thousands of atoms, each of them with their own trajectory, subject to forces, interaction forces between them, and they are usually described with these potentials. And if we need to put an interaction with the electromagnetic field in this kind of description, we need to rely, of course, on Lorentz force, including, of course, Coulomb force. So this is the way. We add another force in these kinds of description. And there are, of course, softwares that can do that. I don't have to write down atom by atom all my molecules. There are databases that are able to make the molecular description atom by atom of these molecules. And there are software, like one is, for example, GROMATS, that is the one that we use, but they are open source. So we can include our Lorentz and Coulomb force in it. Why do we do that? We do that because using molecular dynamics is the tool that we can have in order to see and to follow what biomolecules do and which is their role in the biological system. So, for example, if you follow that yellow sphere, that is an ion that is going through a molecular receptor that is embedded in one membrane cell. So this is, if we observe the different behavior between such a kind of molecule with or without the electromagnetic field in the model, we can understand a lot. Which are the putative targets at the molecular level? Okay, this is basic physics. So what can we think about? We can think about free charges that follow and moves with the electromagnetic field. And in biological systems, the kind of free charge is different to what we are used to think because they are ions. And these are ions, and typically ions in biological systems deal with a barrier. And this barrier is the cellular membrane, because cellular membrane is very selective in permitting ions to go in and out the cell. Each cell controls very well through specific structures that are protein channels what can get in and out. So they move, but when they move, they suddenly find this barrier, and this kind of barrier is at the basis of the special electromagnetic properties of biological tissue, because biological tissues and low frequency has, for example, permittivity values that are up to 10 to 7. That's incredible, but this very large number is exactly related to the fact that here at low frequency we have these double layers of ions that enlarge very much the response. And then, of course, they can move freely inside the space that is extracellular, cytoplasm, nucleoplasm, and so on. But then, don't forget about the dipolar response, because we can find many molecular structures where we found bound charges. and they are typically water. We know that water is a dipolar molecule that oscillates up to 18 gigahertz because it's small and fast. But then there are many other dipolar structures. And one is what we already cited, that are these head of the double layer of phospholipids in the membrane. They are dipoles, but they are also bound, so they cannot do what they want. They can only move a little bit, and then it's full of proteins. Proteins can have a very low, but even very large dipole moment, and so they oscillate. And what is even more important is that depending on their dimensions, each of them will have its own dipolar relaxation at different frequencies. So when we speak about electromagnetic fields, we can think to two main classes of putative molecular targets. One are lipid bilayer, that means membrane, and this is the cartoon, but the molecular model is this, atom by atom, all the head, all the tails, and these are ions, and this red cloud is water. So it's a huge model. And the other kind is what we already discussed, that are these receptors that selectively permit to other molecules to get in or out of the cell. And then you may have noticed that I wrote water everywhere. Water is everywhere in any kind of molecular model of interaction. Water can have a very important role when it is free, so only water, or when it is bound to such kind of structures. And we can have different kind of molecular results. For example, this is the response to this bound water that can follow very well 1 gigahertz. So we can see this kind of changing in the moment, the dipolar moment of water. But also what is very interesting is what happens here. This is I don't know how many of you knows that in the membrane of cells there are channels that are called aquaporins, and they are dedicated to selectively permit to water to cross the membrane, because sometimes cells need water and sometimes they need to put it out. So these are this kind of channels, but these kind of channels can be controlled by or at least the kind of water and forces can be modulated by the presence of electromagnetic fields. So what do we need to do this? We need good molecular models. We need new techniques of analysis. We know this is the era of AI, but this means that we can start to apply big data techniques, analysis techniques that go beyond, because now we have the interaction with an agent, with a physical agent that are electromagnetic fields, and we know how much they can depend on time, and in particular when we speak about modulation speeds. And then don't forget, we always need experimental methods to validate. These are other examples of myoglobin, SOD enzymes. They are all proteins that can be found inside the cells, and they are all subject to an interaction with a possible field. For example, these are bipolar parts that can act on the power density of the molecules. Or here, again, this is the dipole moment of the whole molecule. This SOD enzyme controls oxidative stress. We know how much it may be important, oxidative stress. But then the bilayer, what happens here is now at the frontiers of biomedical applications of electromagnetic fields, it is called electroporation. when a cell membrane is under the action of an intense and short electrical pulses, water can do this magic thing that it penetrates inside the membrane, it opens the hydrophobic tails, and it creates such a pore of water inside the membrane. And this pore of water, what does it do? It can permit to hydrophilic drugs to get inside the cell. This is at the basis of what is called electrochemotherapy, that is nowadays in the sanitary system of many of EU and US states. And we will see more about this. So, as I said, this is a molecular interaction, but then we need to go to cells and we need to go up. So we need to go back to our well-known Maxwell equations, or also, I know that this is a more high-frequency community, but don't forget that here we deal with a very small structure, for example, in a cell. So very often it's, for a given part of the spectrum, is more convenient to work with quasi-static equations where we maintain the dependency of the frequency in the dispersion electromagnetic properties rather than in the description of the propagation. And this is the first thing that we can do, climbing up the scale. We face the cell. And which are the approaches? Okay, it's full of this cartoon. So from this cartoon, we can extract models, continuum models, on which we can do our solution, electromagnetic solution. But then there are also techniques coming from other disciplines like biology. These are images coming from what is called confocal microscopy, and what they are able to do is to focus the microscope at different age. And so they can take images of single cells at different age, something that is very similar to what we know as magnetic resonance. So we can do something very similar with cells. We have all these images. We can elaborate images. We can manipulate with all the kind of computer graphics that we use for medical images, and we can obtain 3D model of cells. And if biologists are good enough, they have many of these beautiful techniques to decorate different parts of the cells. For example, gray here is the cytoplasm, but green here is another intracellular compartment that is called the reticulum, where all the calcium of the cell is stored. And calcium, we will see, it's very important for cells. And blue is, of course, the nucleus. And so when we have this, we can apply the solution of the electromagnetic equations that we think are the best to describe the problem in order to obtain electric field, or this is power loss density, because we use different color. This in blue and red are power loss density in the single part of the cell. Which is the weak point here? The weak point is the continuum description, the electromagnetic properties of the singular compartments of the cell. And this is, from one side, the weak point. From the other part is the bridge domain, because we can, through covariance matrix application, we can derive this kind of properties from the molecular models. We can identify layers for the water, we can identify layers in the membrane, and so on. And from that description, from the dipolar response, the ensemble of the dipolar response in the molecular level, we can extract the electromagnetic properties we may be interested. So that is the bridging domain, and that is also testable experimentally. And of course, cells are not like, they are not exactly like the same, in spite of being same type of cells. And so what we should also be able to manage our stochastic modeling to take into account which are the key features of the cells. And again, experimental methods. Then we keep in scaling up, and we arrive to such kind of this is much better. You can see all the details on how a simple layer of skin can be. We have fat, we have air, we have capillaries, we have gland, we have nerve, we have different level of epidermis that they change on very much their thickness and so on. And so what many people do, that is a little bit tricky now, there is a lot of research in not using exactly this kind of cartoon, but rely on magnetic resonance images, is trying to introduce all these structures inside the layer. Why? The reason is what I said before. We go towards high and high frequency, but we know that in lossy medium, we have this depth penetration that is getting thinner and thinner. So which will be the organ involved in 5G or 6G, they will be skin and eye, so the very superficial organ. So this is important, and again, which is the weak point in following this approach? The weak point is which are the electromagnetic properties of air, which is the electromagnetic property of a sweat gland. And for this reason, many people are nowadays following the approach that I was mentioning, super resoluted magnetic resonance images. But this is on the way, this is the future. And at the end, we arrive where we want to arrive. We want to arrive at the human being. We need to know how to simulate the whole human body, and we need to know the electromagnetic field inside the whole human body. And these are very complex functions because they depend on the shape. So there are what you can see here. This is the virtual population. We have bigger, thinner, younger, and so on. And then there is the position, and then there is the kind of frequency and so on. And then don't forget, I spoke about the fact that we need to validate results on laboratory animals. So we need to have the possibility to estimate the electromagnetic fields also in these animals. So we have what they are called the virtual zoo, and we have rats, mouse, pigs, and monkeys. And then we need to calculate the field. Why? The reason is what we said before, because we need to start to the possibility of interaction and the effect from the molecular level, and then grow up cells, tissue, human body, but it is inside. And it's very difficult to put a probe inside population to know which is the kind of effect of electromagnetic fields you are exposed to when you use a mobile. Nobody would accept to have a probe inside its ear or brain. So we need to have that value inside, and we need to be able to relate it to the field outside. So this is not only animation movie, this is a true approach, inside out. We need to know which is the field that is related to the effect inside, in order to be able to tell to the protection guideline, and not only, also to the biomedical application, which has to be the field outside. And this, of course, in the example of protection guidelines, for example, is that they need to mention both. They need to mention the field outside, that is the most easy value to be tested, because I can come here with my instrumentation and tell you which is the electric field or magnetic field. But if it exceeds the guideline, then the guidelines themselves forecast that you can prove that the field inside is different. And this is the macroscopic dosimetry. And don't forget about the other part, that is the experimental part. So again, we need to look at the biomolecular experiment, but then we have to prove the effects at all levels. And the levels are not, like in the other part, simply tissue, but you have to carry on biological experiments in vivo, and then on human volunteers that are under controlled conditions and so on. So which is the role of the electromagnetic expert, is to give access of good devices that can prove which is the exposure electromagnetic field. Because it's not just a matter of putting a mobile on the top of a cell culture. This would never work, because you don't know which is the field that is in the culture. So this is the work, this is the true work. This is an antenna, loop antenna, adapted to work with animals. These are other kinds of resonance structure. This is a coplanar waveguide, and so on. And these are the requirements for lab experiments. You have to apply all these good practice, biological protocols, of course, but also good practice in designing and characterization of devices. That means numerical and experimental dosimetry in the devices themselves. And then many more, and the one very important is electromagnetic compatibility with the other lab equipment. Because it may happen that you can break the other laboratory equipment, like amplifier of that are devoted to take other kind of signals, or then you can have a sort of crosstalk. So this is all the list of what is necessary in order to obtain fair data and robust evidence, that is findability, accessibility, interoperability, and reusability. And since more than 20 years now, it's well known by who is writing the guidelines for protection that you cannot follow one route only. You need to have biological validation, you have to have engineering validation, and then they need to speak together in order to arrive, to identify which are the proper approaches and levels. And these are some examples. It's strange to think that what is usually a scattering body now is the target of what we want to do. We want to obtain such a kind of electric field or magnetic field on the cells or on the mouse or whatever. And what you have to do is to reconsider how you look at these structures. You want antennas that don't work like antennas. Maybe you want to rely mainly on the near field interaction, because this is what you would prefer. Or also this kind of coplanar structures, they need to emit a little bit, because the cells will be on the top of our guiding structures, and so on. And at any time, you need to perform numerical and experimental dosimetry. Now we'll try to go quickly through some examples of application of what I've said until now only as a methodological approach. And then we'll do it through two examples of projects that we are involved with too. One is related to protection, and the other is related to biomedical application. As I said, 5G technology has reopened the issue of possible electromagnetic effects. And this is mainly due to this new spectrum allocation. So because the other allocations have been already studied a lot. So we move from 900 to 1, but this is the band, and so on. But this is new. So a lot of questions rising, and the most of them were, can we think that exists a non-thermal effect? Because I'm not sure that you all know that until now the guideline protection of human health rely only on the possible thermal effect. But then this power is going down. We have a modulation scheme that is new. So the question now is, can we have non-thermal or long-time effect, and how much can be important to the modulation scheme? And I use the example of what has been done by the European Commission. We have this big call, well-funded, I may say, and with 4 projects selected. But what the European Commission wants is that we speak each other. European Commission didn't want us to take the money and then arrive in 3-5 years and each of us saying a different story. So what we have been, not forced, but encouraged, it was in the call, to do is this cluster that is clue age. And we meet every 6 months exchanging protocols, findings, interpretation, because at the very end, if the results will be different, we need to justify why. And these are huge projects. You can see 20 partners, 22, and with a slightly different targets. In our case, we are involved in this Goliath project. With the projects are huge. The example that I will give you here is starting from molecular, because it's strange that some experimental results suggest that there is the possibility, this is the question, to sense the presence of a high-frequency electromagnetic field also at low power. And the path seems to be the same kind of protein channel in the membrane that is devoted to warm and cold response. So the idea is that it is possible a direct interaction between the electromagnetic fields and these channels in spite of the fact that there is no changes in temperature. And this is what we are doing. We are modeling such a channel, and the channel is quite a little bit complicated, quite a little bit huge, if you think that all these are atoms, and they are not selective calcium permeable receptor, and they are involved in many functions. One of them, of course, is warm and cold sensation. And this is the model you see. We have water. It's a lot of water. These are the number of water molecules. We have all the atoms of the channel. We have the lipids, and we have the ions. And to study if there can be any kind of direct interaction, what we are doing is studying the channel as suggested in literature, and these are the profile of the channel with and without the electromagnetic field. And you can see that here in the central cavity where ions can go through, there is a difference in the dimension. This means that something can be facilitated to get in with the presence of the electromagnetic field rather than without. Of course, this is a super preliminary result, and of course, can you remember the Kitano cycle? We need to prove it. And so the question is, can we prove what are the ions can go through a receptor channel? And the answer is yes. This is the beauty of bioelectromagnetics, that is really multidisciplinary. And exist techniques that are called electrophysiological techniques that are able to measure currents at the level of the single receptor. And they rely on the technique that has quite some years now, that is being developed at the end of the previous century, and they can go down to the level of single pieces of cells, and they are able to seal with this probe the single channels, and this is very interesting. This is the behavior of the current of a single channel. Open or close, you have the current, and you can measure it. But don't forget that we have to measure it with and without the electromagnetic field. And so it's very important that we are able to design and put in place devices that are proper to do that. And these are examples of numerical model. You need to choose the structure, you need to do the numerical model, you need to understand how they want to work. This is a picture of them with the microscopes, their amplifier, their probe, and so on. And you have not to interfere with their work. But you must be sure of which is the power by gram that you are giving to these cells. So you have the part of numerical, again, the Kitano cycle, but also you must put in place the usual method. These are something that you all have seen. These are sensors, these are generators, we have oscilloscopes and so on, but these are thermocameras. But then you have to check numerical and experimental. You see the agreement is good, but we know that measurements are never as clean as simulations are. And then we are ready to go. And once that we have this, in what is called sham and exposed condition. What is sham? It means that I have carried the experiments exactly like this. The only difference is that the power is up. If I change something, biological systems are so sensitive that I cannot put my not exposed sample in the bench far from here. I must put it here, exactly here. But the power must be off. And then we need to move to 26 gigahertz, but again, it's very important that the probe is not affected, because if we affect the probe with the electric field, then the measurement will be affected as well. So the effect will not be reliable. And now we go quick to the kind of biomedical project that is a completely new, different story, that is how we can use electromagnetic fields for regenerative medicine, in particular to restore spinal cord injuries. The idea is to use this kind of device where stem cells are put on it, and using another kind of molecular interactions, that is what I called before electroporation. We know that with electroporation we can create pores in the plasma membrane or intracellular membrane. And these calcium fluxes can control differentiation of stem cells. Stem cells are very important in nowadays research because they can differentiate in any kind of cellular type. And in this case, what we want are them to differentiate into neuronal type. And okay, the idea is that they can differentiate and proliferate, and what is important is that you need, you need to be able to create models of the cells that you will put on your device, but then it's necessary this gap. You need to go to the in vivo to prove the efficacy. And the methods are these. You create the cells with confocal images, you apply your pulses, you verify which is the field. And once that you know, according to the proposed protocols, if the field is effective or not in porating the membrane, then you can tell to biological people that they can go with it. And so these are differentiation examples, sham and simulated. They have been able to quantify differentiation, and sham and stimulated, they have been able to quantify proliferation. So with this kind of approach, you can drive the experimental protocol. And then also what you can do is to grow up at the level of the animal that needs to be exposed, and what you need to do is to recreate the kind of injury that biologists do on the animal in order to know which will be the electric field to which the injury will be exposed. And then you put in place the device, and you can see that the results are very similar in terms of quantities to the ones from the cells. So this is the other kind of climbing up the scale. But at the end, biologists alone, never leave biologists alone when they are with your devices and instrumentation, never ever. So they need to have people like these two girls that are able to control that everything is dealt in the proper way. So these are, for example, measurements of the impedance and the current that were delivered to the animals. These are the animals, quite a reasonable number of animals have been treated. We have not still been able to prove a regeneration because of the timings, but what we have already proven has been anti-inflammatory effect in the lesions, because the responses of inflammation are shorter with respect of those of regeneration. So the very conclusion is that we can follow a unique approach for safety and biomedical applications, given that we are able to follow all the scale of complexity, both theoretical and numerical, and that we are able to interact between the different aspects. And I conclude. Thank you. I spoke too much. I exhausted that.
Noi ci abbiamo provato 100 volte. Andava, poi non va
Thank you
più.
very much, first of all, for the nice presentation.
No, ci abbiamo parlato prima.
About microscopic magnetic resonance imaging. Which microscopic means? What microscopic means? Which is the size of the characteristic that you can investigate?
Well, this kind of there are many different scales down to the molecules presence, because these are what are called the magnetic resonance spectroscopy. So they can go down to any kind of level, and of course this is also a medical imaging technique. So this is really possible to apply to many different kinds of systems.
Any other questions?
So definitely, as it being a party member of the monodisciplinary member, you have multidisciplinary things from nanoscale, mesoscale, macroscale, and then biologists, electrical engineers, and so on, new projects and everything. How much do you have difficulties in understanding the language of your colleagues?
Oh, yes, thank you for this question. I didn't want to point this too much. much, but indeed this is a matter of language that can change a lot. And also the simple concept of low and high frequency, if you speak with a medical doctor that work, for example, in neuronal stimulation, for them, high frequency is 20 Hertz. And the low frequency is 1 Hertz. So the first time that I was dealing with them, I was just understanding nothing. And this is more and more. It's a continuous example. It's a matter of trying to meet and to find a common language together. Yes. For example, they used to wet everything. We do our structures carefully, and they just turn around the cell culture, and everything is wet. They short circuit the signal and the reference electrode. It's complicated, but it's also funny and fascinating.
I have one curiosity about frequencies that are used for, let's say, using electromagnetic field in bone cell construction and so on. There are some, let's say, spectral analysis, or which frequency you use as a function of what? This from engineer point of view, of course.
Yeah, the idea is that these applications usually start from the biological or medical part, and then they come to you. So you have no control at that point. And for example, in the well-known application of bone healing, what they do is they call it low frequency because the repetition frequency is 75 Hertz, but indeed the pulse that they repeat has a completely different spectrum that goes up, that is not a lot, but it goes to 10 kilohertz. And when you tell them, they are completely not aware of this. So you have to take what they bring to you and to make your own electromagnetic considerations. Yes, always. This is typical also.
Okay.
Yes,
sure, but if you talk loud,
okay.
I think we can understand.
I think it's really interesting to see your course full of problems. It seems to be extremely challenging. I used to start up working on antennas, microstrip antennas and so on. And then later on I was exposed to the electronic design and automation industry. They make more complicated things like electronic circuits and so on. But your problems are way more complicated than those that I face in EDA, for instance, the electronic design and automation industry. Are there some low-hanging fruits for electromagnetic people like me we can break into, because these problems are just too way complicated for us?
I must say that, of course, they are more complicated because biology is more complicated, but I must confess in front of you that you have much less control. So you have much less control of what you can do with antennas or electronics or circuits, because you can never be totally sure on how biology will respond. When you deal with biology, things, standard deviation increases, and everything is less under control. The only point is that you find a good multidisciplinary team. If you can rely on your biological colleague, then everyone can do that if interested in specific applications. There is no need to have the control of everything. What I presented here comes from many years of collaborations, interactions, learning languages. But when you can rely on the biological or medical counterpart, then you can do it because you are the technological counterpart and they would rely on you.
Other curiosity or... Okay, so thank you again.
Thank you very
much. And then we can go for the last part of our meeting.
It's a matter of
usually for the airport.
Grazie comunque del